Now, the second stage we want to talk about is we want to talk about the last process of pyruvate production, and we'll move on to the next stage. Now, outside the mitochondria, we have a fermentation process that usually happen. So pyruvate is a pivotal metabolite in a cellular respiration. What does that mean? This is what we need to move to the next stage. So if we have oxygen, if O2 is not available in the cell, there will be a process called fermentation. So the fermentation process would take place within the cytoplasm, not in the cell, not in the mitochondria. So if O2 is available, what would happen in the cell? The cell will make the pyruvate to go into the mitochondria, and it's going to happen to produce aerobic. So if there is no oxygen, this will happen in the cytoplasm. So this is the fermentation process. If there is oxygen, this will happen in the mitochondria, and this is the aerobic respiration process. So let's talk about the fermentation process. The fermentation process is an anaerobic process that reduces pyruvate to either lactate or alcohol and CO2. So the things being produced after the fermentation process is either lactate or alcohol with the CO2 being produced as well. So what happens is NADH would transfer its electrons to the pyruvate. So the pyruvate is the CCC we had before. So now NADH, which uses the electrons and hydrogen ion to produce NADH from NAD plus before, it's got energy, would transfer the electron which you use, which is only one electron, which was used before, which is two electrons, which was used before, is going to transfer it to the pyruvate. So alcoholic fermentation carried out by yeast produces carbon dioxide and ethyl alcohol. So the alcoholic fermentation, which yeast usually do, produces these two products. So this has been used in the production of alcoholic spirits and also used in production of bread. So at the point in time when the bread gets hotter, so the alcohol would evaporate from it. So lactic acid fermentation also is carried out by a certain bacteria, which is a lactobacillus, and a fungi, which produces lactic acid or lactate. So this is being used in the production of cheese, yogurt, and sauerkraut. So we also have other bacteria, which is used to produce chemicals anaerobically, including isopropanol, butyric acid, propionic acid, and also we have acetic acid, which is being produced by these bacteria. So now let's talk about the process of fermentation. Now this is almost similar to what I've explained to you before. So in the process of the breaking down of glucose, so we had 2 ATP being taken and one is being broken down. So the phosphate will attach to the G3P to energize the process. So this is going to give you G3P, 2 G3Ps are being produced here. So what happens is at a point in time, 2 ATPs want to be made. So our 2 energized phosphate molecule will be added to the process. So that gives you what? That gives you 2 BPGs. So mind you, the energy which is already the electrons which has been released and the hydrogen ion released in the process of the breaking down of the glucose would be used to produce NADH. So this is going to be the production of NADH. So what happens afterwards is the BPG will not produce 4 ATPs. So to leave only these carbon atoms, which is going to be the pyruvate. So now if there is no oxygen, so the process continues to produce lactate or alcohol. So, and this is being produced by animal or bacteria, and this is being produced by plants and yeast. So this is a summary of fermentation process. So now let's move on to advantages and disadvantages of fermentation. So what are the advantages of fermentation process, which is the process that is being carried out when there is no oxygen? It provides a quick burst of ATP energy for muscular activity. So it takes a longer time for the production of ATP during the cellular respiration. But in case you don't have it and you don't want to carry out some activities, so there will be a quick production of it using the anaerobic process. So when muscles are working vigorously for a short period of time, lactic acid fermentation provides ATP. So what is the disadvantage? Lactate or alcohol are toxic to the cell. So these are very poisonous because they're acidic. They're not very okay for the cell. So lactate would change the pH, would drop the pH, and cause the muscle to fatigue. So this is why you feel pain in your muscles when you really work hard. So this is because oxygen depth, you don't have enough oxygen for the process, so you feel the pain. So yeast die from alcohol that produced by fermentation. So people might say, oh, how do we have yeast in the body? Or how do you have yeast in your food? The yeast is dead because the alcohol being produced will make the place acidic. And yeast do not really, they don't thrive in acidic environment, so they easily die. So efficiency of fermentation, you know what efficiency is the meaning is how effective is that thing. So 2 ATPs are the only ATPs produced during the process of fermentation, when this is going to give you a total of 14.6 kilocalories. That is a total kilocalories produced from 2 ATP. But when you have a complete oxidation of glucose, which means the cellular respiration is going to produce 686 kilocalories. Now, how do you know the efficiency? So what you do is you say 14.6 divided by 686 times what? Times 100%. So if you calculate this, let me use my calculator quickly. It's going to give you, give me 1 minute, please. So 14.6 divided by 686 would give you 0.021. So when you multiply by 100, 12, so that would give you 2.1%. So this is the efficiency of anaerobic respiration. So the meaning is the amount of ATP produced during the amount of energy you get during the process of anaerobic respiration, which is fermentation process, would only produce ATP, which is equivalent to 14.6 kilocalories compared to 36 to 38, which you produce during the cellular respiration. All right, so let's move on to the next stage. So this is a summary of what we've said. The input is glucose. So in this process, you've got two ATP being produced. So the fermentation process would not lead to the whole production of 2 ATP net. So, and mind you, you also have lactate and alcohol produced in the process of fermentation. Now let's talk about what happens in the preparatory stage in the mitochondria. Mind you, I told you this before. Some textbooks or some scientists, they don't refer to this preparatory stage. They move directly to the Krebs cycle. But I would talk about this in case you find in an exam where you have only 3 stages, just remove the stage and pick the glycolysis Krebs cycle and electron transport chain reaction. So, but if you have 4, then you use this as a number 2. So the preparatory prep reaction connects glycolysis to citric acid cycle. So what happens is there is this is exactly, so it's a preparatory stage for the citric acid cycle, which is another name for Krebs cycle. So the end product of glycolysis, which is pyruvate, would enter the mitochondrial matrix. So the pyruvate is converted to 2-carbon acetyl group. I told you the way it happens. So we have 3C, which is pyruvate. So one of the carbon is being used to produce oxygen, because it's being used to produce carbon dioxide, and you've got how many remaining? You've got out of these 3 minus 2, 3 minus 1, you've got 2 carbon remaining, which is called acetyl-CoA. So there is a specific enzyme, coenzyme A, which is which is going to attach facilitate the reaction to produce acetyl-CoA. So electrons are being picked up as well, and hydrogen atoms. So hydrogen atoms have been released, and electrons have been picked up. So, and to produce NADH from NAD+. CO2 has been released and it's been transported out of the mitochondria into the cytoplasm. So this will be expelled from the cell. So this happens 2 times per glucose molecule. So this would happen 2 times per glucose molecule. And this is why we have 4 ATP. being produced. So, and this is the process. The pyruvate is this plus these the coenzymes, CoA enzyme. So produces two acetyl-CoA and carbon dioxide. So this process, because it happens two times in the it happens two times in glucose. So this is why you've got two NADH. So the quantum mechanism, the preparatory stage will produce for you two NADH. As I understood, it's going to produce for you two NADH. It's going to produce for you acetyl-CoA. It's going to produce for you carbon dioxide. So these are the things produced in that stage. So now let's talk about the mitochondrial structure and the function. So we've already talked about the two membranes, which is got the inner membrane, it's got the outer membrane. So we've got the space in between the membrane. So it's called intramembrane space. We've got the matrix. So this is the place where the second and third stage would take place. And we've got the cristae. So the cristae is these places. These ones are called the cristae. So this is a location for electron transport chain reaction, ETC. So the matrix is a location for the prep reaction and the citric acid cycle reaction, or the Krebs cycle. So now let's talk about the citric acid cycle process. So it's also called the Krebs cycle. This happens in the matrix of the mitochondria. This begins with the addition of C2, which is acetyl group, from the acetyl-CoA to a C4 molecule. So you have mind you, you've already had a C2, which is the acetyl-CoA. So what happens is it's going to be added to the C4 molecule. So there will be a C4 molecule, four carbon atoms, which is called oxaloacetate. So the oxaloacetate is actually a C4 molecule. So this is going to take the C2, which we had before, and form a C6. So this C6 molecule is called the citric acid. So what happens is the NADH and FADH2 would capture energy-rich electrons. So they capture the energy-rich, the electrons which are very energized, they capture them. So what happens is they use that energy to produce ATP. So the ATP is being formed by a substrate level phosphorylation. Anytime you hear the word phosphorylation, is a process of production of ATP, or a process in which we use a phosphate to produce something. So let's call it phosphorylation. So it twice of one glucose molecule, once for each pyruvate. So this will happen two times. So it happens two times for one glucose molecule. So that is to say, it's going to happen in 4 times total in the whole process. So this is why it says 4 CO2 is being produced, because every pyruvate produces one CO2. And since it's happening 2 times for one glucose, and one glucose has got what? It's got 2 pyruvate. It's got 2 pyruvate, and it happens 2 times. So that means it's going to be what? It's going to do, and one pyruvate produces one CO2 and one CO2. So if you multiply this by this and this by this, you're going to have 2 CO2 and 2 CO2, which is going to give a total of 4 CO2 at the end of the process. And mind you, the energy-rich electrons will be used to produce 2 ATP. And we have a production of 6 NADH. Why do we have 6 NADH? Because every process, each of these processes we have in the Krebs cycle will produce 2 NADH. And 2 NADH being produced in 3 places would produce 6 NADH. You got 2 FAD per glucose molecule. We're going to talk about that in more detail in the next slide. So now let's take a look at the procedure of citric cycle. So in the citric cycle, which is here, what usually happens is, in the process here is a summary of all the procedures that happen. So in the glycolysis stage, so what happens is a conversion of glucose into pyruvate and the creation of reduced form of nicotinamide adenine dinucleotide. So this has been reduced here. So that is why you see NADH has been produced. So this is what happens in the glycolysis. But at the end of it, what happens is you've got 4-ADP. I mean, 4 adenine diphosphate converted to 4-ATP. So this is what happens here. So you have 4-ADP converted to 4-ATP by adding 2-ATP and to the process, to the procedure. So giving you 2 net ATP. So the process will give you 2 net ATP. So the preparatory stage creates a reduced form of nicotinamide adenine dinucleotide. So with 2 ADP is being converted to 2 ATP. So why do we have this? Because this has been reduced already by using the electrons and hydrogen ion in the system. So in the citric acid cycle, it creates a reduced form of nicotinamide adenine dinucleotide and flavin adenine dinucleotide. So this is the process in which flavin adenine dinucleotide has been involved. But in the preparatory stage, it only produces NADH. It did not produce FADH2. So there is the citric acid cycle that actually produces the FADH2. So, and during that process, it goes to the next stage, which is the electron transport chain. So electron transport chain and the process of chemiosmosis would help to convert 32 or 34 ADPs to 32 or 34 ATPs. So this is a summary of what we have in the slide. So now let's move on to the next stage, which is a citric acid cycle. So in the citric acid cycle, we can say the C2, the C2 acetyl group will combine with the C4 molecule to produce citrate. So a C6 molecule. So you can say this is a preparatory reaction. We got acetyl-CoA, the 2 carbons with CoA, which is the enzyme. So the enzyme is CoA here, will not go into the reaction. It only helps to facilitate it. So then when the C2 gets into the reaction, they combine with a C4 to form a C6, and that's called a citrate. So what happens after the citrate is being produced? So the second stage is going to be the oxidation. So the second stage is the oxidation reaction. So the oxidation reaction will produce from NAD plus is going to add with 1 hydrogen ion, which is in the system, is going to form NADH plus another hydrogen. So this is how then in the process of producing 2 NADH plus hydrogen. So it's going to release 1 CO2. So mind you, CO2 is going to have one of the carbon from the C6, isn't it? So now it reduces it to C5. So the loss of the 2 CO2 will result into a new CO4. So this is going to happen 2 times. So it's got one being left and another one is going to leave. And mind you, this one is going to convert to produce NADH plus hydrogen. So the oxidation reaction produced 2 of this. So 2 of NADH plus H plus is being produced during the oxidation process. So this is the process that takes off the one that leads to the production of CO2. 1 carbon from this citrate with oxygen, because it's an oxidation process, is going to combine with it to form CO2. So now the third stage, the loss of the 2 CO2 results in a new C4 molecule. So because I told you before, you're going to lose the CO2. It's going to happen 2 times. So that would lead to, this is the second one. This is first and this second one. So that would lead to a C4 molecule. So the C4 molecule, after that is been got, then there is a stage for production of ATP. So 1 ATP is being produced by substrate level ATP synthesis. So the meaning is the substrate level ATP synthesis is these are the ATP that needs to be produced during the process of the reaction. So this is not that where does the ATP come from or where did it go. ATP is a currency of energy. The energy in the system would actually use, not all the energy is going to be used at the same time. So they need to measure the amount of energy being put into every system. So this is why 1 ATP is being produced here. So now the next stage is going to be the production of FADH2. So FADH2 is being produced by the additional oxidation reaction. So additional oxidation reaction from what we had before will produce FADH2. As you know, the oxidation reaction will need you to have electrons and hydrogen ion being added to the wall, being added to FAD plus. So that produces the FAD, sorry. So that produces FADH2. So, and when that is being produced, another NADH plus H2 is also being produced. And what happens afterwards? So additional oxidation reaction will produce FADH2 and another NADH plus H2 and would regenerate the carbon-4. So once the carbon-4 molecule is being regenerated, it's going to continue again into the procedure of the reaction. So it's going to join the C2 here, and the C2 plus the C4 will form the citrate again, and the procedure would continue. So this is a summary. So how do we remember this? Because this is a big question. How do we as students remember this? The way to remember this is simple. The first thing to remember is remember that you've got acetyl-CoA and you've got the which is C2 plus CoA enzyme. So the C2 enters the citric acid cycle. It combines with the C4. So someone would ask you, where did the C4 comes from? So the C4 molecule is in the system. So once it gets into the system, it combines with it gives you what? C6. So the C6 will go through a process of oxidation. So the process of oxidation would lead to the production of NADH plus H plus, and would get off one carbon dioxide. And the process happens again a second time. It gets off another carbon dioxide. When that happens again, another NADH plus H2 is being produced. So that leads to the formation of what? The formation of C4. Mind you, you've got oxidation happening 2 times. So that is why you've got loss of carbon, 2 carbons, making it to have what? 4 carbon. So after that process, a production of ATP is being generated. So one ATP is being produced. And once one ATP is being produced, it helps to energize the system and gives an additional oxidation, which produces FADH2 and NADH plus H2. So if we count the total number of NADH2 produced in the system, you've got 1, you've got 2, you've got 3. So this is why at the end of the reaction, you will have a 3 NADH plus H plus. So we're going to count everything at the end of the reaction. So then, mind you, this is only one acetyl-CoA that got into this. It's only one. And also this is got only one, only one FADH2 being produced. So I'm going to show you what is going to lead to after this. So now we want to count how many are being produced. So let me go back to my slide so that we don't get confused. So as you know, we got 1, we got 2, we got 3 NADH plus hydrogen being produced, and we've got only 1 FADH2 being produced. This is only a reaction for one of the pyruvate. One of the acetyl-CoA. So one of the acetyl-CoA, and mind you, we've got 2 acetyl-CoA from what? From the glucose. So what would happen is you multiply this by 2. So this by 2 and this by 2. So this leads to a total production of 6 NADH plus H2 and 2 FADH2. So this is what we're going to see in the next slide. So the next slide says, in the input, what you put into the reaction is 2 carbon acetyl groups. So then the output, what you produce from it was 4 carbon dioxide. I think you understand how you got this. So we got 1 being produced. So we lost 1 carbon. So we got the other one being produced. We lost second one. So that is to say 1 acetyl group produced 2 CO2s, isn't it? So because this is one of these, this thing happened 2 times because we've got 2 acetyl groups. So then you multiply this by 2, that gives you 4 CO2. So also NAD plus into the reaction is giving you 6 NADH. Then we've got 2 FAD giving you 2 FADH. So then we've got the ATP being produced. It says 1 because it's only 1 we looked at. So that procedure of Krebs cycle happens 2 times. So it leads to the production of 6 ATPs. So this is a summary of the process. So now let's move on to the electron transport chain.